This is the recipe for life and how tRNA fragments fit in. All living organisms are made using a simple recipe. Genes are transcribed from DNA into RNA, which is translated into protein. Think of this process like cooking a meal. DNA is the ingredients in your mac and cheese recipe: noodles, cheese, flour, cayenne, mustard, milk or water to boil. RNA is the prepped and ready to cook ingredients: grated cheese, boiled water, mixed dry ingredients. Protein translation can be thought of as the steps you take to put all the ingredients together into a meal, a delicious mac and cheese. Traditionally, scientists have studied diseases like cancer by trying to understand DNA mutations that cause large shifts in cells. Think of this like swapping out angel hair noodles for the macaroni noodles in Grandma's Blue Ribbon mac and cheese. At the end, you might get a gooey yellow hairball instead of some scrumptious mac and cheese. In our lab, we study subtle differences that can cause disease. We focus on transfer RNAs that you might think of as plain as the water used to boil your mac. The water doesn't add to the flavor of the final dish, but it allows you to prep the ingredients and put it all together. It turns out that cells can reuse transfer RNAs to regulate the process of making DNA into RNA into protein. Think of this like saving some pasta water to thicken your cheese sauce with. We've observed this process in a number of different human cells and diseases, and we are working to understand when and why cells might save some of that starchy goodness to perfect their mac and cheese.